Let's start our special talks. The first one in the special talk series is today Arkasubhra Ghosh from Narayana Nethralaya Foundation, Bengaluru.
Good afternoon, everyone. Thank you, India Bioscience, for this opportunity to talk. I come from sort of a halfway house between an academic institute and a clinical institute. So we are a translational biology unit. Essentially, I did my bachelor's and master's in microbiology, and then went on to work on molecular medicine and gene therapy. So currently, I am a member of a variety of different uh, committees in the government. Somebody had spoken earlier about lots of different committees in the government. So I'm a part of a lot of those committees that specifically look into the area of cell and gene therapy. One of the key things that we've contributed to in the country is, of course, write the national guidelines for cell and gene therapy in India. That's the first one that came out. So who we are is essentially at the core tertiary care hospital. So many years ago, back in 2012 to be exact, Dr. Rohit Shetty, who is the current director of the hospital, he asked a bunch of questions to me, asking, we have a lot of therapies, but still I have a subgroup of patients who respond poorly. I don't know what to do for them. They keep coming back. And then there's a bunch of patients for whom we have no treatments. So what can you do? So he said, let's build a lab. So how are we going to build that lab? So right from the beginning, the aim was to do translational research, right? So in order to do that, the discovery itself has to be from the patients. So therefore, we set about building a large biorepository with clinically annotated samples and associated follow-up clinical data. So using electronic medical records, et cetera, and then started building up the molecular capabilities so that you can have an integrated analysis done to define exactly what are the driver mechanisms of the disease, how can you intervene, etc. So eventually get to the point of bespoke therapies. So genes. So most of us here work on genes at some point. So we end up publishing a lot of papers like these. So these are papers from our group and as part of direct papers or consortia, etc. So what do all of these kinds of discoveries mean in the clinic? So it's what the patient may have, right? You discover a bunch of mutations in a particular phenotype, and then you define that disease, you define the course of it, so on and so forth. But the problem still was for a lot of genetic diseases, and that's where this talk is more going to be focused on. We do not really have therapies for it, right? So genetic testing is there. And over the past 10 years, when we first started a whole genome sequence, a whole exome sequence, it used to take 70,000. Now it only costs about 7,000. So it's becoming more accessible, more equitable. So more and more people can avail those services and know what's wrong with them. But we still have to provide solutions. So that brings us to the rare disease epidemiology, and that's essentially what I want to do in today's talk. All of you brilliant folks, if I can impress upon you that this is a field that needs more heads, needs more experts, needs more people to actually do things for these patients. So there are about 7,000 odd rare diseases, but of which only about 450 are reported in India. So you can imagine for such a large population, there's a lot of underreporting that's happening, right? So there's a lot of research that needs to be done in just identifying, describing, etc. Then, of course, you have, so this is just from the National Rare Disease Policy. You have three different groups of it. One's able to transplant, so they can be treated. Ones for which lifelong treatments are available, and ones for which definitive treatments are either not available or exceedingly expensive. Right, so all of this is very expensive. And that's why there's around the world a lot of interest in precision biotherapeutics, right? So currently there are about 48 products that are available, FDA approved, but there are more than 2,000 trials ongoing. However, in India, in the rare disease area, there aren't any, right? So we need to fix that. And why it should be of interest to you is because the government is interested in it. So across the agencies, now there are about 10,000 crores earmarked for funding in this area. So if you are attempting to set up a new area of research, this is it. Come to the rare disease area. So these are examples of gene therapies, particularly AAV-based gene therapies. AAV stands for recombinant adeno-associated viral vector. It's the gold standard for gene therapy vectors around the world. But what you can see that across blood disease, muscle disease, eye disease, motor neuron disease, everything is in millions of dollars, right? So clearly, it's not equitable. It is not accessible. And of course, none of them are made in India, so they're not available in India also. So our lab was set up to address this particular gap. And we work on these major areas, tissue engineering, regulation of expression, vector production, and then, of course, regulatory processes as well. So I will briefly show examples of these, but to talk about our journey, essentially, when I can set up the research lab back in 2013 within our Nethralaya, the idea was to have this patient-directed research. So when Dr. Shetty asked all these questions, what to do, then we sat and thought about it, and we planned this this pathway of discovery and identification of targets, etc. A key component that needed to be fit in was the patient, right? The patient involvement. That, I think, is what started for us and really brought us into this arena properly, is engagement with the patient community. So that started back in 2016. So by that time, we had the biology and there was some proof of concept, etc. But then you need to then move to an actual therapy. So for that, you needed to have a production facility. You needed to go through the regulatory processes. So that's essentially the journey that we were on. And just for the crowd here, how did we get all this funded? So back in 2015-17, we had these little grants from the Karnataka government called the VGST grants. So these were only 20 lakhs. So for all of those who are starting careers now, it's very nice because earlier on, the starting grant was 20 lakhs. Now it is 2 crores under ICMR, right? So life is good now. But of course, you have to get it. It's still competition. There's no doubt about that. So we started from that. But we work with a lot of these patient communities. So this is the muscular dystrophy community. This is the GNE myopathy community. And then we were able to get a DBT Wellcome Trust team science grant for about 10 crores. So that was a handy grant. We went on to continue working with more and more of these patient trusts and startups as well, and went on to the stage now where we have big grants like ICMR CAR grants, which are like 15 crore grants. So those, as well as larger international and industry grants, which fund this large endeavor. But we still need even more for scaling up production for the large patient base that we have. We have more than 100 million patients, right, across different diseases. All right, so I'm going to take an example of Duchenne muscular dystrophy. This is a fatal disease, affects boys only, X-linked genetic disease. The mutation happens in this protein called dystrophin that is an important structural protein of the muscle. So because of its mutation, your muscles eventually degenerate. You stop being able to breathe, the heart stops functioning properly, and you die eventually over a period of 20 years. So the work in our lab essentially started because of these two kids and their mothers, actually. So this is Dhara and Jyoti. So they are warriors. So they came, met me at several places. They met many people and sat in our lab essentially till we agreed to start a DMD program. So thankfully, even though we are an eye hospital, our chairman is very open to all kinds of research and all kinds of endeavors. So he was happy to support the program initially. so that we could get started. So how are we going to do that? You have to build up the technology as well, right? So in order to build up the technology, you have to have the transgene designs done, you have to have capsid designs done, formulation, production, etc. So it's a long process. And we can't just say that we are going to create a therapy and then say, oh, we've specialized only in the molecular biology of it, only transgene design, because that doesn't help the patient. And that's where a lot of the problem happens. So that's why you need to collaborate to build more. So in our lab, we went on to develop these transgene designs. So what you are seeing are these are just myoblasts. These are early myotubes. So you can see them aligning themselves into these myotubes. So these are from 2 different DMD patient lines, and you can see them having difficulty aligning themselves. So, but when you do a therapy on them, they start turning into these elongated cells, they start fusing, and we developed all of the different technologies around it, the microdystrophin, mini dystrophin, exon skipping, etc., to evaluate which ones work better for which kinds of patients. Oh. Oh, okay. All right, so essentially to make a choice of what kind of vectors we should work on is why we developed all of these technologies side by side, because in different patients, different therapies might be of use. And there's an important aspect also. So here it's a single vector, here you have double vectors, and these are hyper personalized. So this is only for a small number of patients. So therefore, you can't really use the same kind of therapy for everybody. So now I'm just going to show you. So that was a choice that we made, that for the first human clinical trial, we are going to go with the microdystrophin. But then we went on to do what is called lead optimization and de-risking. So we tested out many different, so this is essentially transgene engineering, protein engineering, in order to see which domains work better, etc., etc. So this is all the science part of it. And then we went on to work on these different capsid mutants to see which mutant expresses more. But what's really the application of a capsid mutant? So now it's time for everybody to wake up, watch some mice running. We have all eaten food, so better to watch mice running. Now, what you will notice here is that the first one is control. This has got no therapies, and that's really not able to run quite so well. And you have these different mutants at low dose and high dose, and you can see that they run differentially. What you will see in so in order to do so this on the run on the treadmill, they have to continuously generate force, and that is the problem in the DMD patients as well. They're not able to generate force. So after 20 minutes, you can see that the wild type capsid ones, they are not able to run anymore, only the mutant ones run. So essentially this shows us that we have reduced the dosage by tenfold. So that's one step towards equity, right? So we have reduced the dosage from what would be required. So therefore, now we want to take this into trial. So now we come to regulations. So in order to do that, we need to have counseling, we need to have the patients, you need to make a choice of the therapy, etc., etc. But there were no major guidelines for it at the time, right? So in 2019, the guidelines came and we contributed to that. And we continue to have our troubles with the regulatory, because the regulatory bodies also do not know what to do with these kinds of trials. So you have to work with them. So that brings us to the topic of resilience. So we are making things in the lab, we have to make it bigger. A 10,000 times jump. How are we going to do that? So we can't sit around and say we can't do it. We have to make it happen because nobody else in India is doing it. So we went on to build a clean room facility. Again, lots of people's input in order to build it. We built all the SOPs around it. We built all the technologies around it. And this is called a fixed bed bioreactor process. It's very boring. Nobody would be interested in it. But at the end of this process development, we were able to make vectors about 5 times to 10 times more. So essentially a cost reduction. So that's the whole point of it. So I jokingly tell my postdocs here, they published a small paper out of it, but the paper actually cost us more than 2 crores just in reagents. Okay, so that's the effort that needs to go into it. So in order to do all of this, so how are we going to make this equitable? So we are going to have indigenous technologies. We are going to have locally held IP. So all vectors, all production processes, everything is ours. So we do not need to pay $1 million license fees to anybody. We have reduced the dosage to be given per patient, and we have increased our production scale. So put together, I think we can now bring this therapy for 1/30th to 1/50th of the international cost. So that's the aim. Now, in the final story I have to tell you is the power of collaboration. All of this doesn't happen just by myself. It's a very large group of collaborators we have in the country and around the world. So this is Devajyoti. He is best known for having the gene editors. And together, we were trying to cure a rare disease patient. This was an N of 1 trial that we were trying to do. And he made the editor, and I made the vectors. And this is the timeline for it. We started in October, and we had collaborators at NYU and Rome. And by September 23, we had developed the therapy, tested it in animals, tested it in the patient-derived iPSCs, and we were ready for saving this patient's life. However, just around the time when we were doing it, the patient passed away because of complications. So we couldn't reach the end goal, but it pointed out one important aspect that I want to tell all of you is work with the patient community. The patient community is coming forward in India in a big way, trying to support cutting-edge research, which are difficult to find traction in government agencies. So please work with them. There's lots to be done. So this was featured in Nature as a story from India. All right, so this is what we have done. I'm gonna end with this slide. So develop some good habits, have a big picture, frame your ideas, build networks, keep writing grants, come to the rare disease community, write grants for it. There's lots of money in it. I'm sure you will all get good grants for it. All right, so with that, I'll thank everyone. And if anybody is interested in joining our group, welcome. Thank you.
Thank you, Arka. Questions?
Yes.
Can we have the volunteers, please? Can you please raise your hands again?
Thank you for the talk, sir. I have one question. You have published so much papers in Nature. I wanted to ask, how is the review process in Nature? What do they expect when compared to other journals?
Oh, they expect the moon and back. So it has to be original. You should have proven it in 7 different orthogonal models, and it shouldn't have been published by anybody before. And the authors should be stalwarts in the field. What else? What else can I list?
How much times more intense than the regular journal?
So that's easy. So if you like have a journal of your field, usually somebody else was also pointing out that the top journal of the field is usually between 3, 5, 7, something like that. So in ophthalmology also, it's like that only. So you should take 5 of those journals, the amount of data that you would put in, and put it in one paper, and that would be it.
Thank you for the clarity.
Hello, sir. Wonderful talk. I'm Shrangika Soni. I'm postdoc at CCMB. So I have 2 questions, one scientific, one non-scientific. So I just wanted to know how difficult it was to get a funding for rare disease. Because
So as you can see, I told you, back in 2015 to 17, in that time, I only was able to get 20 lakhs for it. Okay. Now, I can't write any more grants because I've got so many grants. So that's the difference, because at the time, and it's a huge difference, it's a huge shift. We are at an inflection point, I tell you. All of the government agencies, they are looking towards personalized medicine, precision biotherapeutics, not just for rare disease, for everything. okay, for all diseases. You come up with cell and gene therapies, mRNA therapies, vaccines, antibodies, etc. in this space, excuse me, in this space, and it's going to be pretty well funded. So this shift happened from 22 onwards. I think the mindset change happened post-COVID. Till then, it was not as dramatic. And even from the regulatory standpoint, that's when everything opened up. And from the patient advocacy point of view, I think a lot of uh, uh, points went into that. Probably nobody noticed, there's this thing there called DST MOTA. So MOTA stands for Ministry of Tribal Affairs. So I have a sickle cell grant because the Ministry of Tribal Affairs is funding the sickle cell programs in the country. So I'm a beneficiary of that.
One more, because we are also working in gene therapy and stem cell therapy in spinal cord injury. So how did you maintain your titer of AAVs in bulk production?
Oh, yeah. So that's the whole science behind it. So like I said, the 2 crores that went into figuring out how to get the titers up. So it was a DOE experiment, right? Design of experiment. So you have these multiple parameters. So you examine each parameters in isolation and then in combination and then come up with the whole process of it.
Hello, thank you, sir. Great talk. So, sir, I'm a bit curious about knowing what are the key challenges in running clinical trial for either a Ayurvedic medicine or a normal allopathic medicine. So, what are the key challenges of clinical trials?
So, for Ayurvedic medicine, there is no challenge. Your only challenge is whether your patient comes back to you. Okay. That's the only, frankly, that is the challenge. For allopathic medicine, there's a whole horde of challenges, right? So usually, depending on the condition, I mean, it's true even for the well-defined diseases like genetic diseases where you have a single mutation driving the disease. It's very hard to get patients with a very strict set of phenotypic features and which the patient has not been treated for. Okay, so for example, if you want to run a diabetes trial and you want somebody who has not taken X, Y, Z different medications because that may be confounding your final results, it is usually very hard because in India people take a lot of medications. There's Ayurveda, there is the local homeopath or Babaji or something will always be there, right? So all of that confounds trials in India. The biggest problem that we have is patient non-compliance. So the patient is supposed to come, let's say, at 6 weeks for their next visit or intervention. They'll come anytime between 10 to 12 weeks or not show up at all. So for large observational studies that we have this study on diabetic retinopathy. We have 7,000 patients in it. And we needed 3 years of follow-up on it. We had only about 1,500 completed follow-ups of the 7,000 patients. So you imagine the amount of enrollment that you have to do in order to cover however many patients you need to do. Now that problem is of course limited in something like CGT, but for other medication types, it is a problem.
Thank you, sir. Thank you.
Thank you for the interesting talk. So I have a question broadly on the national mission of cell and gene therapy.
Mm-hmm.
So given I'm more interested in establishing my lab more on the clinical front with the therapeutics, so the question is that there are a couple of therapies which are approved recently in the West for the immune checkpoint inhibitors. And then in case I want to continue the work, so those therapies are still not approved in India. So are there any policies which are coming up? So the therapies which are approved by FDA, so the timeline can be shortened for approval.
Yeah.
And
it's already there.
Okay.
It's already there.
So how much would roughly be the timeline, or it would be easy to
of course,
use this?
it's context dependent, but anything that is approved by the US FDA or EMA or Japan, that goes into what is called the fast track. So you can get a license to actually sell it, and you can do post-marketing surveillance on it and provide the data, depending on how it has been approved. So, for example, for cell and gene therapy or even for rare cancers, you have the N of 1 trials, and there are approvals given on the basis of N of 1, N of 3 kind of trials. So, they have to undergo a different process compared to a proper phase 1 to 3 trial that a regular drug does.
So,
Yes.
under the rare diseases policy, the government has set aside up to 50
50
lakhs
lakhs.
per patient for every rare disease patient for their
Yes.
treatment. Would gene therapy also be
Yes,
covered under that?
it would be.
So, in
the
some
disease
sense,
has to be in the schedule. That's the only thing. So that is
Right.
another sort of fight going on.
right. But that would then be, in some sense, there's already a captive
Cap-
market.
correct.
Right? Once you clear all the regulatory hurdles, the market is assured, even though
Assured
it's
for you.
rare,
Yes.
the government is paying for the
Government is
treatment.
paying for it.
So
Absolutely.
That's also just
That
to
is a
share
huge
with
incentive.
people. It's a huge
Yes.
incentive to work on that.
Absolutely.
Wonderful talk, but I wanted to understand what is the licensing around the CRISPR usage in gene therapy here in India?
So Debo holds and IGIB holds the license for the FN-Cas9 throughout the world, even in the United States.
Okay.
So while the two folks there were fighting it out, he in the meantime got the patent even in the United States. And the current generation of the vectors have higher specificity than the Cas9s.
So just to add to that, sorry, there's no regulatory issue with CRISPR like there is with transgenics.
Mm-hmm. Yeah, that's right.
Interestingly, even for crops.
Even for crops, yes.
Thank you. We'll take later.